kívánok! Hölgyeim és uraim, tisztelettel köszöntöm Önöket a Magyar Tudományos Akadémiai mai rendezvényén, amikor sor kerül Kovács Sándor 2022-ben megválasztott tiszteleti tagunk székfoglaló előadására. Kovács Sándor 9 éves volt, amikor 1956-ban a családja elhagyta Magyarországot. Ezért aztán azt írta egy levelezésben, hogy szókincsem nem egy Magyarországon iskolázott és itt felnőtt képességével rendelkezik, ezért azt kérte, hogy a székfoglalón a bemutatása és az előadása angolul hangozzék el, egyébként pedig magyarul beszélünk. Úgyhogy engedjék meg, hogy ismertessem Kovács Sándor szakmai életútját. Professor Sándor Kovács is founder and director of the Cardiovascular Biophysics Laboratory in the Department of Internal Medicine's Cardiovascular Division at Washington University in St. Louis since 1990. He completed his undergraduate education in 1969 with a BS in Analytical Engineering at Cornell University, his MS in 1993, and PhD degrees in Theoretical Physics in 1977 were obtained from Caltech, California Institute of Technology. His thesis, titled The Generation of Gravitational Waves, was supervised by Kip Thorne, 1997 Nobel laureate in physics. But as I've read, there were many other excellent physicists at the same time in the department with whom Professor Kovács was in close relation. Just one famous name, Richard Feynman, who also received the Nobel Prize in 1965. He obtained his MD degree after 2 years of intensive study in University of Miami's highly selective PhD to MD program in 1979. His training in internal medicine and fellowship cardiology were completed in St. Louis at Washington University Medical Center, where he joined the faculty in 1984. He is board certified in internal medicine and cardiology and is professor of medicine with joint appointments in physiology, biomedical engineering, and physics. He has served as PhD and MS degree thesis advisor to dozens of students in physiology, biomedical engineering, and physics. His international reputation is well established by his election in 2006 as president of Cardiovascular Systems Dynamic Society, an international society for cardiovascular physiologists, biophysicists, and cardiologists. He is a recipient of the Master Physician Award from Barnes-Jewish Hospital Medical Association, and I came across another must be prestigious award called Öcsi Bácsi Award in Caltech, and I'm very curious to learn what Öcsi Bácsi Award is. He was elected member of the Hungarian Society of Cardiology in 2012, and for his contributions to cardiovascular sciences received an honorary doctoral degree from Lund University, Sweden in 2018. In St. Louis, he has hosted visiting faculty from Budapest Heart Center and has co-authored several publications with Hungarian colleagues. He served as ambassador between 2007 and 2020 to Budapest University of Technology and Economy as part of Washington University's McDonald International Scholars Academy, and he remains Ambassador Emeritus. He has lectured at Semmelweis University, KFK, BME, Lund, Stockholm, Oslo, Paris, Tokyo, and throughout the United States. He has served as editorial board member and/or reviewer for major journals in cardiovascular physiology and medicine, such as American Journal of Physiology, Circulation Research, Nature, etc. Professor Kovács's main interests involve an interdisciplinary framework involving the physical, biophysical, and clinical sciences. He has pioneered the direct application of methods of classical physics and mechanics to solve problems encountered in clinical bedside cardiology, particularly the issue of diastolic function of the heart and its quantitative assessment. He has numerous patents related to methods for mechanisms-based diastolic function assessment using bedside echocardiography. His ideas and methods are currently in use in Lund and Karolinska in Stockholm. Based on his collaborative international contributions that have advanced the field of cardiovascular biophysics and physiology and clinical cardiology, he has been elected as honorary member of the Hungarian Academy of Sciences in 2022. Today we have the privilege to listen to his inaugural lecture, which entitles The Science of Diastole. So please, Professor Kovács, deliver your lecture.
Jó napot kívánok. Even though I'm supposed to speak in English, I couldn't resist. I want to thank all of you for coming to listen to my inaugural presentation. And with that, we'll get started. There may be some members of the audience to whom the word diastole is a new word. And so I took this from Wikipedia to remind me and you of where it comes from and what it means. It's from the Greek, meaning to dilate and spread apart. It refers to the filling phase of the heart. If you now go to the Google Ngram viewer, which is a search engine, and you can type in whatever word you like, in whatever language you like, it'll plot for you the appearance of the words that you're searching for as a function of time in the world's total digitized literature. And so you can see, if you type in diastolic function, Doppler echocardiography, nothing happens until 1980. I mean, nothing detectable. And then all of a sudden in 1980, there's this rapid upstroke with a dip and then a continued upstroke after 2000. In red, you see Doppler echocardiography, which came into its own in the early 1980s. So the fact that this upstroke happens at the time that I started doing research in cardiology is a complete coincidence. I am not responsible for the upstroke, but I contributed to the upstroke by publishing papers. So with that as a historical introduction, I'm going to do a very quick review for the non-physiology, cardiology, anatomy members of the audience, so they understand and have some conceptual framework by which things happen. The usual anatomical description of the heart in books, in color, is shown here, where, as usual, red is the arterial blood, and blue is the venous blood. And what you see in the front here, you can read the labels themselves, but the point is that the most anterior surface in the chest of the heart is the right ventricle. If you then remove the heart from the chest, which all of you have seen in Raiders of the Lost Ark, when the priest, by this hot furnace or something, reaches in and removes the heart from a poor member of the group and holds it up, and it's beating in his hand. Of course, they don't show you what he left behind. And what's presumably left behind is this structure, which shows that the heart is suspended in the back by the 4 pulmonary veins and is anchored on top by the great vessels. There are no other adhesion points or rivets or fasting mechanisms. This was known to us in books, but actually it was relatively recently clarified, recently meaning ancient history, by Leonardo, who understood the structural relationships of the intact heart and of the great vessels. And you can read the captions yourself. And in further detail, if you look carefully here, Leonardo shows you the closed aortic valves viewed from above and the open aortic valves. There are 3 leaflets, and they kind of form a triangular opening until they fold completely back. He also understood the role of the papillary muscles. Let's see, I'm trying to move it, and how they relate to motion of the valve plane. All of this will become a little clearer as I proceed. In addition to this, the most important development came in 1628 when William Harvey published this book, which proved that the blood circulates in the body. People weren't sure. People thought the blood came from the liver, and it was used up by the spleen. And even though I'm sure they saw arterial injuries with pulsating blood, it never connected that it's a closed circulatory system. Here's the example that Harvey used to prove which way the blood flows in the venous system. It's kind of obvious if you think about how it's being done. Currently, the way we teach circulation is by the usual cartoon, with blue blood being venous, red blood being arterial, and the blue blood comes in, gets oxygenated, it turns red in the lungs, and is pumped to the rest of the body. Now, when you look in more detail to the actual anatomical components of the heart, they're the usual. Here we go. There we go. Right atrium, right ventricle, left atrium, and these parachute cords are the things that prevent the valves from flipping up into the atrium when the ventricle is pushing blood out the pulmonary artery. But not only are there mechanical components, it turns out that the heart is electrically active, with the pulse being generated by a bundle of cells in the atrium going down toward the base and then spreading out through both ventricles to result in the mechanical contraction of the heart that we see. And when the heart empties and fills, this is the method by which it's taught in medical school, with blue blood coming in through the open tricuspid valve into the right ventricle and then ejected in systole out the pulmonary artery, and oxygenated blood entering the left atrium, going into the left ventricle, and then being ejected out the aorta. So here is a 3-dimensional MRI data set in which a method of analysis of the data is used, which is called pixel following. And you can plot and color code the initial location in space and time of every pixel in the image, and you can follow it for one cardiac cycle. It just repeats again and again, and it spins around in 3 dimensions so you can appreciate what happens. As you're sitting here in the audience, this is happening in your chest. You feel nothing, because you're not supposed to feel anything, but you can appreciate that depending on the method of analysis, you can extract a tremendous amount of information about the heart works as a pump, both in systole and diastole. And the color code, I need to spend just a little time on this. The color code is the red ejection is into the aorta in systole. The 2 yellows are blood coming from the lung into the left atrium, into the left ventricle. The blues are the inferior vena cava and the superior vena cava that mix venous blood prior to it entering the right ventricle. And there are amazingly informative details just by looking at the pattern in which the blood flows and swirls. For example, your inferior and superior vena cava that allow blood to come into the atrium are not aligned, they're misaligned on purpose, because what happens is when the blood comes in, it spins the blood. And by generating angular momentum, the rotating blood tumbles into the right ventricle, requiring less energy for the right ventricle to fill. The other thing that is not obvious, but can be gleaned from this, is that the left atrium, which is filled with the two yellow shades, kind of yellow and mustard, that there is yellow and mustard blood leaving the lung, entering the left atrium, both in systole and in diastole. So there's continuous filling throughout the cardiac cycle of the left atrium. Even though the heart's beating, the atrium is filling twice. Once during ventricular systole, and then again, when the ventricle fills, another filling wave comes in. For cardiologists, this is our review. For most other people, this is something they've never appreciated or had an opportunity to think about. So now this forces us, or allows us, to use a conceptual framework in which we view the entire behavior of the four-chambered heart both as a pressure pump and as a volume pump. Volume pumping means you take the blood at low pressure and you kind of move it along. And based on the numbers and the function, 3 of the 4 chambers are volume pumps: the right atrium, the right ventricle, and left atrium. And the left ventricle is a pressure pump in systole and a volume pump in diastole. Right. The LV is only a volume pump in diastole. So how do things look? So because I'm an invasive cardiologist and I do cardiac caths, this is a pigtail catheter sitting in the left ventricle of a patient doing a cine angiogram. And what you can see is the catheter comes up from the femoral artery. It crosses the aortic valve, which is here. And the way you cross the aortic valve is you wait for systole. And when systole occurs in a cardiac cycle, you quickly slide the catheter past the open valve. Go into the ventricle, and then you pump. What that showed, as you saw, was the ventricle filling and emptying with contrast. Compare the angiogram with the cine MRI on the right, which is a single slice of the four-chambered heart, showing the left ventricle, the right ventricle, the interventricular septum, and the 2 atria. This is, again, a cardiac MRI slice of the heart, perpendicular to the four-chamber slice. This is the short this is how you slice a loaf of bread. You take the longitudinal stuff and you slice it perpendicularly. And so this is the ventricle happily beating. You can see that the blood pool in white gets smaller in systole and gets larger in diastole. And you can appreciate that the outer surface of the ventricle doesn't move very much. And if you look at the next cycle, this is a peristernal long axis, cardiac MRI cine loop, the same pumping action except in a different view. There we go. On the short axis view, I was hoping to point out that when the ventricle contracts, it actually twists like this. And that twisting is explained by the orientation of the muscle fibers throughout the wall of the ventricle, so that the outermost fibers go in one direction, the middle fibers go circumferentially like the belt around your waist, and the inside fibers go up and down. And the contraction of all these fibers causes the ventricle to thicken and get smaller and eject blood. And remember that muscle is conserved. So the same amount of muscle you have in diastole is the same amount of muscle you have in systole, except it changes shape. Now, if you want to take a look at the fiber orientation in real life, these are cardiac cine loops of anatomical dissection of a dog heart, showing the orientation of fibers from outside to inside as you move through the wall. And on this one, you again see it's a different display format. It shows both the left and right ventricles as you go from inside to outside. And when you wind up at the end, right here, you can appreciate as you look through the wall that the fibers go from one direction through to the other continuously. If you want to get a better sense of the volume pumping function of the four-chambered heart, you can get that information from cardiac MR by taking short axis pictures like you see in frame A, and then digitizing the contour and stacking it up like you see in frame B. If you plot those cross-sectional areas, what you see is that the total volume of the four-chambered heart dips a little bit in systole and then regains its full four-chamber volume in diastole. And the total amount of the dip compared to its overall volume of maybe 900 mils is about 5%. So if this line were completely flat, it would be a perfect constant volume pump. The little dip in the volume curve shows you that it's a constant volume pump to within 95%. And you can display this by making a movie out of it, and you can see when systole, this is the pericardium, this is the outer surface of the four-chambered heart. And if you look on the bottom here in systole, you see dip a little bit. Cis, and that's it. So for those of you who are MDs and who are required to read plain chest x-rays, when you put up a chest x-ray and you look at the cardiac silhouette, you're looking at how big it is relative to the thorax. But you never think about what phase of the cardiac cycle the chest x-ray was taken in. And the reason it doesn't matter is because the four-chambered heart is essentially the same volume in systole and diastole. Even though inside the ejection fraction may be 70%, the outer silhouette doesn't change much. When I asked that question as a medical intern, I got criticized for wanting to know too much. So when we put all of this together, we can conclude that the four-chamber heart is a constant volume pump. Now, the best way to illustrate physically and intuitively how this constant volume pumping and reciprocation of atrioventricular volumes occurs is to take a look at the pumping action of a 4-year-old normal child. And now you can see that when the atrium gets small in systole, when the ventricle gets small in systole, the atrium gets big because the atrioventricular valve plane is moving up and down. It's reciprocating. That's one view. Here's another view, the four-chamber view, and you can again appreciate that the atrioventricular valve plane, which separates the atria from the ventricle, it's a teeter-totter. And another, a parasternal view, which shows you the reciprocation of atrioventricular volumes. By focusing on this volume pumping feature of the ventricle, you have no choice but to appreciate that constant volume pumping, that the longitudinal displacement of the valve plane must be a valuable index by which cardiac function can be quantified, not just subjectively, verbally, but by measuring the displacement in millimeters and the velocity. Here's a cartoon, an idealized version of constant volume pumping in which the ventricle is the colored bowl. The thickness of the wall is physiologically appropriate for end systole. The dimensions are physically appropriate. And on the right, you see transmitral Doppler, which I'll talk about in a minute. And so this is how, ideally, the left ventricle moves when it fills. It has a fixed outer surface. The muscle thins, and as the muscle thins, the annulus moves up. This certainly helps to explain what happens. What happens when this method fails? Well, here's a pathological example in constricted pericarditis where the annulus is prohibited from moving up and down, but the wall still contracts. And so it should be physically intuitively obvious that if you can't accommodate volume this way, you can only do it this way. And that's exactly what constrictive pericarditis does. If you take a look at how this moves, you can see that the annulus barely goes up and down longitudinally, and you can see that the outer wall of the ventricle is being tugged and it's pulling on everything on the outside. If you look at the other view, you see the same thing, which is that the annulus, which you saw in a 4-year-old child wildly go up and down, is restricted from moving, and the only way the heart can accommodate the volume is by external displacement of the outer wall. So this is just a good example of how nature can compensate for pathological scenarios. So what are the constant volume consequences? Remember, in the video we showed the red blood, blue blood, yellow blood. You sort of intuit that what should happen is when the ventricle ejects and blood leaves, blood comes in at the same time. And that is exactly what happens with the pulmonary veins. And the point that I made earlier is that the atrium fills continuously through the cardiac cycle. There are 2 filling waves into the atrium. Once when the ventricle goes into systole and pulls down the mitral valve plane, and then again when the mitral valve opens and additional blood enters. So the 5% deficit is due to the difference between the red blood leaving the aorta and the simultaneous yellow blood entering the atrium. Those 2 volumes are not equal. They're off by 5%, and that's made up in the next diastole. The appreciation of this volume conservation physiology is in part due to our efforts to teach cardiologists how things work. So I'm showing you this again because this is the cine loop where you can appreciate that the red blood going out the aorta is replaced by the 2 yellows coming into the atrium. But if you watch the yellows coming in from the atrium, there are 2 waves in the cardiac cycle. And that's the point I was trying to make, that the left atrium fills continuously. The swirling is, I mean, for those of you who feel comfortable with fluid mechanics, if you have blood moving linearly in a straight line and you bring it to a stop, the pressure rises because you release energy. The way to avoid pressure rising when blood stops flowing is to convert linear momentum to angular momentum. So you spin the blood instead of bringing it to a physical rest. These different colors have allowed us to characterize the functions of the atrium more accurately than is usually appreciated. And what it shows is that the constant volume physiology framework teaches us that the usual atrial function indices are actually generated by the ventricle. And I'm going to go into the woods a little bit right now and look at a 2-chamber view of the left atrium and the left ventricle. If you imagine it, think of it as a glass cylinder with a washer with a hole in it. It's sealed at both ends, it's got juice. And what happens is, how do you relate the velocity of the washer going up and down with the velocity of the fluid through the hole in a constant container? And the constant volume physiology makes a prediction. And the prediction that it makes is that the velocity of blood going through into the ventricle through the mitral valve, which is called the Doppler E wave, and the simultaneous velocity of the annulus going in the other direction. So remember the movies? In the movies, what you saw was the valve plane going up and down. It's obvious that when the ventricle when the ventricle is in diastole and filling, the annulus is rising. So the annulus is going up, but the blood is going in. So there are two opposing velocity directions. That's why these graphs, one of them points up, the other one points down. But the prediction is that the ratio of these two things should be constant. And if you do that, what you get is it's a constant for the first 100 milliseconds, and then it takes off. And so it proves, this in vivo data proves that constant volume physiology, annulus going up, blood going in, is a constant, it generates a ratio in vivo in a real heart as predicted by the primitive cylindrical view of things. But you can go further, and you can explain why the ratio is related to the end diastolic pressure, which is what this data shows. Cardiologists use that ratio clinically to estimate filling pressures of the ventricle. And what this paradigm shows, that you don't have to make a guess, that there's a causal relationship between the ratio and the end diastolic pressure. This line of thinking of volume, cross-sectional area, and filling allowed us to publish a paper in Nature in which we showed that the cross-sectional area of the atrium and the cross-sectional area of the ventricle, when approximated in simple geometric terms, the difference in the areas generates a force. And that force contributes to filling of the ventricle in diastole. And that's in the graph here. But my point in showing you this particular slide is that the volume pumping conceptual framework is what leads to this kind of analysis and new insight about how the heart works when it fills. So this was all volume pumping. Now it's time to talk about the pressures and flows. This is a graph of a paper we published a while ago of transmitral flow by Doppler echo. This is velocity as a function of time with simultaneous pressures in the atrium and the ventricle. And what this shows is that when the flow accelerates, mitral valve opens and the flow velocity goes up, there's a pressure gradient where the pressure of the ventricle is lower than the pressure in the atrium until you get to the peak. And then the fluid decelerates, the velocity goes down, and simultaneously the pressure gradient changes sign, S-I-G-N. And so what this reveals is that after the mitral valve opens, the ventricle starts filling very rapidly, but while it's filling, the pressure in the ventricle continues to drop, which is counterintuitive. You would think that if you fill a container with fluid, the pressure should go up. That's not what happens in the heart. So the behavior of this system allows us to characterize it as an inertial oscillation and model it like this. So your ventricle is made of muscle, it oscillates. But if you want to oscillate right here in the room and go like this, you use two muscle groups, extensors and flexors. So when you contract on this side, you relax on this side. When you expand, you contract the muscle here and relax here. The heart does not have an opposing muscle group and it still oscillates. So it's a reasonable question, how does it achieve that? It's a single muscle. So what we conclude based on this is that the simultaneous drop in pressure and increase in volume requires that the heart is a mechanical suction pump at the instant of mitral valve opening and for a little bit thereafter. So it literally goes and sucks the blood in from the atrium. There's a common misconception in physiology and cardiology, which is that the heart can only be a suction pump if the pressure in the heart is less than the pressure in the atmosphere. That's incorrect. Because the source of filling for the ventricle is the atrium, not the atmosphere. And so it's relative to atrial pressure that the pressure drop matters, not relative to the atmosphere. This has generated an exchange of editorial viewpoints in the literature over the last 20 years, but I think it's finally been agreed upon. When cardiologists do echocardiograms, which all of you have heard about, and they measure and they plot the flow of blood velocity across the mitral valve, they see these E waves and A waves. E is for the early filling, and A is for the atrial kick. And you can see that the patterns, all of them have E and A waves, but the shapes of the waves are different. In general, they all roughly look like triangles. They go up and down, but the details are different. And simultaneous with the transmitral flow is the velocity of the mitral annulus, which is called E prime and A prime, which goes in the opposite direction. The question now is, these are the things we see. We know that the heart is a suction pump. How can we explain the shape of these waves quantitatively? And again, we're back to this constant volume pump, suction pump framework. Well, I have to put in Newton's law, F equals ma, and when you write it algebraically, it has three parameters for this primitive analog. It's got a resistor, an inertia, and a spring. And those are and how far the spring stretches. And those are the three parameters that describe this motion. And the prediction is that if you write this equation and you record an E wave in transmitral Doppler like this, in echocardiogram, and you use the E wave contour as the points to be fit by the solution to the equation, then you get the green line. Not only do you get the green line, you get the parameters of the equation. In other words, you solve the inverse problem, which is you give me the E wave and I give you the parameters. And what falls into your lap at no charge is a new observation, which is that when you look at the mitral annulus and the E prime wave, depending on the patient, you see it oscillate up and down, up and down. And in general, cardiologists have not paid very much attention to it, but we have shown that in people in whom the annulus, look, this is ringing of the annulus. The velocity goes like this. So it's that ringing that tells you what the properties of the ventricle is. And it's good to have the ringing. If the ringing goes away, then you have what's called relaxation-related diastolic dysfunction. So the ventricle is not springy enough. Now come the obvious critical questions from reviewers and others, and smart students, which is, okay, you've got this spring mass thing, it's supposed to be the heart. Gee, when I look at the heart, I don't see a spring. Well, the question is, what is the analog of the spring? And the answer is known. It's partially the extracellular matrix. So if you dissolve the muscle cells in the ventricle, what's left is a sponge where the muscle cells used to be. It's the marbling in the meat. And that is elastic. And you can envision that it's got some equilibrium configuration, and if you deform it, it'll spring back to its equilibrium resting state. This was first appreciated in 1986 in this paper by Robinson and Factor and Sonnenblick. So that's the extracellular spring. Here's the intracellular spring, which is the giant protein titin, T-I-T-I-N, which stores elastic energy. And the experiments show that if you, titin has an equilibrium point. If you stretch it, it comes back. If you compress it, it comes back. And if you stretch it, it comes back. So in the physiologic range, which is from here to here, it's a linear relationship. So the protein tighten is a bidirectional spring. If you compress it, it pushes you back. If you stretch it, it pulls you back. And it lives inside the myocytes. So it tells you that there's an optimal length for resting cells. But there's more. Totally by accident and unintentionally, people at the NIH were trying to take pictures of the electrical circuitry on the surface of the heart using calcium imaging. And they discovered that if they peel off the visceral pericardium, then when the ventricle is relaxed and slices, it springs open. What that means is that the heart itself has shrink wrap. And the shrink wrap stores elastic energy, and in diastole helps to expand the heart. This was a completely accidental discovery. So when it comes to the spring in the model, it includes all these anatomical, physiological components. So that's the spring. What's the dashpot? Well, the only choice we have for the dashpot is the relaxation phase of the heart. And if you think of systole as a cramp, it's the ability to relieve the cramp internally by calcium sequestration inside the cell. But there's more. It turns out that myocytes have an extensive have a membrane network called the T-tubule network. And if the T-tubule network within this is a picture of this is one heart cell, which looks like a brick. And these are tunnels that run in and out XYZ, Cartesian-wise, in the cell, and they basically contribute to the simultaneous electrical depolarization of the cell, so that when the cell contracts, it contracts everywhere at once, as opposed to like an accordion. Pop, 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 pop. And look at the trouble nature went through to assure that cellular contraction, it's simultaneous by building this network. And I'm going to try to wrap up by asking the obvious question, since we talk about stiffness and relaxation in these models, are those two things independent or do they depend on each other? And we've shown recently that they do depend on each other, and you can quantify numerically the relationship between stiffness and relaxation by the asymmetry of the Doppler E wave, upstroke versus downstroke. And even if you remain skeptical, you can ask, what is the proof that a simple spring model like this actually, that the K parameter is actual chamber stiffness? It's a legitimate question. Here's the answer. You do a cardiac cath, you put the catheter in the ventricle, you measure simultaneous change in pressure, change in volume, and you relate it to the simultaneously obtained transmitral echo E-wave derived K parameter, and you get an R of 0.84. Which for in vivo routine physiologic relationships, it's quite convincing. And now I'm going to finish with this, which is intended to get your eyes to glaze over. But the words are easy. We published our idea in 1987. And here's the oscillator, the spring, and all you need to look at is this red thing. And the red thing says that the velocity is some constant times a negative exponential times a hyperbolic sign. That's all. That's the contour of the E wave. In 2010, friends in Norway came up with a different model. It has 6 parameters, not 3. And their result for the velocity of the E wave is a constant times a negative exponential times a hyperbolic sign. It's exactly the same relationship that they derived using a completely different line of argument, a much more complicated model, which, by the way, is not invertible. I can't give them the E wave and have them come back and give me the 6 parameters. In this model, you can give me the E wave and I can give you unique values for the 3 parameters. So it gave us the confidence that we had the right idea. So what does this all reveal in a summarizing sense? Number one, all hearts are mechanical constant volume suction pumps, pretty much, within 95%. In order to fill, the ventricle, the muscle must relax. As far as the load is concerned, the heart chambers and muscles are designed to, whatever load you can present to them, they will handle, because if you overload them too much, then they will prevent you from breathing by going to pulmonary edema. Relaxation, stiffness, and load are the three parameters that uniquely determine diastolic function. And our method, which is called the PDF method, is the right method for it to do that. And so with that, I conclude my presentation and show you pictures of my family for whom I express heartfelt appreciation. This is my dad here, and my mother. They're both deceased. This is Katie when she was a baby. Katie's right here. And son Sándor, my sister who's here in the audience, wife Diane in the audience, daughter Katie, and son Tom. And so now this progeny generated this, which is Joey, who's in outer space right here. He's 4 years old, and Maya. They belong to Katie. Rhea is 18 months, and she belongs to Tommy and his wife, Nicole. And Adam and Zoe belong to son Sándor and his wife, Shanna. And I also want to acknowledge the people from whom I have learned, who are in part responsible for me being here. I put down Kip Thorne because he was my thesis chair. Feynman, all these people were on my thesis committee. Fowler won a Nobel Prize in '85, Feynman in '65, and they were all on my thesis committee. And I'm not going to tell you what questions they asked, because they're tricky. At the University of Miami, Eric Rees was the person who paid particular attention to my intellectual and clinical development. And in St. Louis, it was David Kipnis, who was the chairman of medicine in those days. And I want to acknowledge my students and the funding agencies who supported my work over the years, as well as my collaboration with the Lund Cardiac MR group, from whom some of these cardiac MR loops were provided. And I would like to express my appreciation to the membership selection committee, some of whom members I've never met, but I've seen their names. And with that, I conclude my presentation. Thank you very much.
Köszönjük szépen ezt a nagyon szép és érdekes előadást, amiben talán a legfontosabb, meg talán a legszebb az volt már legalábbis számomra, hogy egy multidisziplináris megközelítéssel hogy lehet még mindig ilyen abszolút alapfunkciót felfedezni a szívben, amiről az ember azt gondolná, hogy ott aztán már mindent tudunk. Nagyon-nagyon szép előadás volt, nagyon köszönjük, professzor úr, és az én dolgom, mint osztályelnök most már csak annyi, hogy átadjam a tiszteleti tagságról szóló oklevelet, ami ebben az esetben kétnyelvű, magyar, de nem angol a másik nyelv, hanem latin. Úgyhogy we all are better off if I'm reading it in Hungarian. A Magyar Tudományos Akadémia 195. közgyűlése Kovács Sándor professzor urat az elméleti fizika és az orvostudomány kutatása terén kifejtett kiemelkedő tudományos tevékenysége elismeréséül a Magyar Tudományos Akadémia tiszteleti tagjává választja. Gratulálok, professzor úr, nagyon sok aktivitásban, egészségben eltöltött, és még sok sikert hozó évet kívánok a tudományban, és nagyon sok örömet ebben a szép családban. Gratulálok!